Once again to be introducing the Barshop seminar series speaker, and this week we have Dr. Robert Shmookler Reis. You're right. Okay, so Robert joins us from the University of Arkansas for Medical Sciences, UAMS, in Little Rock. He wears several different hats at the moment, and I think starting with Professor of Genetics, Biochemistry and Molecular Biology, and Pharmacology and Toxicology. I guess they're joint appointments. He's also a senior research career scientist at the Veterans Medical Center there in Little Rock. And he's also an endowed chair, which is the highest a sort of recognition given to faculty there. So those who know Robert will know also that he's perhaps one of the pioneers in the aging research field. And I think it's fair to say that he was probably there from at least the early 80s, and I think was marked the very beginning of the aging field. And in that light, he is well known for his work on quantitative trait loci mapping and using QTLs to be, in fact, the first person to map longevity associated genes. And he used C. elegans and then mice. I believe he moved to a mouse model looking for aging and age-related genes. Out of the mouse work came some interesting genes that affected osteopenia, and that was translated into humans. One of the other genes that came out of that worm work was REC8, and I was very surprised to hear that many years later, that was actually found to be one of 5 or 6 genes that was positively selected for longevity in whales, 200-year lifespan. So other contributions that Robert's made extend to his most recent studies that we're going to hear about today on neurodegeneration and finding proteins that are involved in mitigating the aggregated proteins. And we're going to be hearing about CRAM1. This work was recently published. So I've been trying to get a hold of Robert to come down here for the last couple of years, so I'm really glad that he came. I just want to finish by saying that I first met Robert about 14 years ago, and that was at a worm meeting in LA. And what I recall was this guy that just kept asking these pointed and questions that just reflected a great depth of understanding. And I'm very glad to report that after 14 years, I was at a meeting there last year in Madison, and he's still delivering those same kind of pointed questions. So it's very good to see.
Thank you very much, Shane, for that very kind introduction and for the invitation here, and thank you all for coming. So today I wanted to tell you about the most recent and most exciting avenue our work has taken to try to get a worm's eye view of neurodegenerative diseases and the aggregation of proteins that precedes and triggers that. So the first point I want to make with this slide is showing aggregates from a variety of different neurodegenerative diseases. Huntington's disease here and here. This is a Russian military laser, so if it blinds anyone, let me know. But I'm told that if the recorders are getting past the part where they're supposed to stop, I can blind them with this. Alzheimer's disease here, Parkinson's disease here, and ALS, amyotrophic lateral sclerosis here. And you'll see that they all present aggregates that can be detected by a variety of means, including antibody to ubiquitin or antibody to specific proteins that were first recognized as initiating these aggregates, and I call those seed proteins. I came into this thinking it's too much of a coincidence that so many neurodegenerative diseases feature aggregation, foci, bodies, whatever you want to call them, in the cytoplasm or nucleus, for that not to be a hint of a common mechanism. And so I went into, I approached this from C. elegans because it has, it's a wonderful model system for aging, but it also provides us with some very instructive models of a variety of neurodegenerative diseases. So we have models, several models for Alzheimer's, several models for Huntington's disease, and Parkinson's disease as well. So we started with a strain from Rick Morimoto that expresses Q40YFP. That's a string of 40 glutamines in a row fused in frame to yellow fluorescent protein, which in the larvae is diffuse in the cells, but by the day 2 adults, even by day 1, you see aggregate foci forming, and by day 5 adults, those are brighter and more numerous. Interestingly, the aggregates that form are very, not quite evenly distributed, but they're distributed between cytoplasm and nucleus. So most of the neurodegenerative diseases form their aggregates exclusively in one or the other. Huntington's is exceptional in that it forms aggregates both in nuclei and in different individuals in the cytoplasm. And in fact, that's what we saw in this model. So the green is the YFP. The orange is a false color for DAPI, so that's showing nuclei. I'm sorry, I've got it backwards. The DAPI is green, and the orange is showing the YFP. But these yellow foci are nuclei that also have YFP. So you're getting a mixture of the two localizations. So obviously, aggregates will form where the aggregable proteins are most abundant and most susceptible to bumping into other aggregable proteins that they can coalesce with. And this is just a brief summary of some of the neurodegenerative diseases: Huntington's, Alzheimer's, frontotemporal dementia with Parkinson's-like features, Parkinson's itself, ALS, and prion diseases. And you'll see that of these, only Huntington's is nuclear and cytoplasmic, but Alzheimer's has extracellular A-beta plaque, and also, as I'll show you, some intracellular A-beta, and tau is cytoplasmic neurofibrillary tangles in the neurites, in the projections of neurons. But frontotemporal dementia, cytoplasmic, Parkinson's, cytoplasmic, ALS, cytoplasmic, prion disease, nuclear. So they're all over the place, but this is a model for both types of aggregates forming. Not sure why. So we focused on expression of one of the two proteins that's shared by several diseases. Actually, there's more than two. There's tau, which is often hyperphosphorylated. There's TDP43, which was first identified in ALS, I believe, and then also found in frontotemporal dementia and Parkinson's. Ubiquitin and ubiquitin-like proteins are found in all of these categories. And I guess that's all I pointed out. Alpha-synuclein is found in several of the diseases as well, although originally in Parkinson's. So the protocol that we use to prepare ultra-pure aggregates, we perfected in C. elegans. Well, perfected implies that we got to perfection. So we optimized or we improved in C. elegans. And we start with worms that are either raised in a normal way or with a RNAi to knock down expression of a particular gene. We froze them and ground them in a very small mortar and pestle appropriate to worms. We did a lipid extraction to reduce peroxidation chain reactions, did a quick differential centrifugation, and then we immunoprecipitated with antibody on magnetic beads, either to GFP in the case of the Q40 YFP, or it cross-reacts, or with antibody to A-beta or tau in the case of the Alzheimer's samples, which I'll get to towards the end of the talk. And then the aggregates that you pull off here are carefully rinsed and isolated, and then dissolved or resuspended in 1% sarcosyl with beta-mercaptoethanol, which will redissolve some fraction of the aggregates, but not others. So we separate the aggregates into what are called the insoluble, sarcosyl insoluble and sarcosyl-soluble fractions. And these are then dissolved by near boiling in Lemley buffer and run on 2D gels or submitted for tryptic digestion for proteomics. And what I wanted to show you here is this is what the aggregates look like that we've taken off the beads. They look about the same size as the aggregates that we saw in the worms. And this is the same amount of the rinse-off that did not have aggregates, same equivalence of lysate that didn't bind to the beads. And if we had the lights completely off, you could see that there's a faint green background here. It's not important enough to ask for the lights to be dimmed, because the only point is that, and it's all right, the point is that that's the unaggregated Q40YFP, which still also binds to the magnetic beads. But it's not isolated as these aggregates, so we get rid of the diffuse stuff when we do the centrifugations. So the first thing we did is 2D gels to just sort of look at the sort of material that we're capturing. And we isolated these at day 3 and day 5 from wild-type worms. And in the sarcosyl insoluble fraction on the left and the sarcosyl soluble fraction on the right, there's an increase in each of these fractions in the amount and complexity of the protein that's isolated. So we ran 1D gels, sliced them up, and sent the individual slices for protein digestion and analysis by LCMS. And we got lots of GFP, as you might expect. We got a total of 59 proteins that had a false discovery rate of less than 1%, so we're confident in the identifications of those proteins. And we found 9 proteins that have Q-rich repeats. And these include PQN53, these stand for prion-like, there's a family of these programs, quite a lot of them in C. elegans, 2220. ATX2, which is the seed protein for one of the ataxins, ALX1, PIS1. These proteins all have Q-rich repeats, which can become extended. We got a couple of proteasome subunits. We got a couple of neuropeptide precursors. We got 5 proteins that had been implicated in neurodegenerative diseases. This one we called CRAM1 because we didn't really believe it was HYPK, but HYPK was what got us interested in it. It was by homology weakly but significantly homologous to Huntington-associated protein K. Ubiquitin-like protein 1, PGAM5, HSF2, ATX2 all appeared as well, and 6 proteins that are implicated in aging, including the FOXO factor, DAF16, the competing nutrient-sensing transcription factor, PHA4, DAF4, DAF12, Werner's 1, RS kinase, and WE1.3. And so we tested these proteins in a way that is facile in C. elegans but difficult in any other system. It's one of the benefits of working in C. elegans, by doing RNAi knockdown. So we looked at the number of aggregates per worm, an easy thing that you can just count, at an age where there would be 70 normally. And if we knock down this HYPK CRAM1 protein at the RNA level, we lost in number about 45% of the aggregates. And we did slightly worse, but still significantly rescued the aggregation phenotype with this QN-containing protein PQN22, PQN53, ATX2, and UNC108. So these were all interesting candidates, but we're going to pursue them more or less in order of their functional impact on aggregate formation. And I think we will need the lights down for this because you can just barely see. Well, I guess you can see. You can see that there are aggregates here in the mock siRNA. I have control of this? Okay.
It's over here.
I think somebody did it before you. I'm not sure. Anyway, it's good. Thank you. Okay. So you're getting aggregates in the mock siRNA, but in CRAM1 knockdown, you get no signal at all that you can see, and in PQN53 you get a little bit. So you can either count the number of aggregates in blue, or you can score the intensity of the aggregates per aggregate in orange, yellow, whatever that is. And both go down about 55, 60% with CRAM1 siRNA and at this particular age. PQN53 has a lesser effect. But here's the interesting thing. If you look at the decline in total aggregates, multiplying this decrement in blue in count by the decrement in intensity per aggregate, you get 86% reduction in aggregate content when you knock down CRAM1. But it only made up 1 out of 200 hits. Okay, so it's contributing roughly 0.5% of the protein in aggregates, and yet it has this huge effect on the amount of aggregate that you get when you reduce it. On the other hand, PQN53, which made up a much larger percent of the aggregates, 27% of the total hits, had a proportionate effect on the total amount of knockdown. In other words, it's like PQN53 acts stoichiometrically. The amount that's there, it's gone, and so there's that much less aggregate. Whereas CRAM1 is acting in some kind of a leveraged way.
Robert?
Yes?
So one possibility there is that this was mass spec data, right?
Right, right.
Is that those peptides didn't fly.
Is that what?
There was a particularly protein that just didn't want to fly in the mass.
So mass spec has that bias that I use that term percent of hits very cautiously and loosely. Maybe the two are in conflict, but I should put inverted commas around it, quote marks around it, because yeah, some proteins simply will not generate a peptide after tryptic digestion that you can identify. So, but when you're comparing things, then it is meaningful if it's the same protein compared to state 1 to state 2. But when you're saying percent of total, take it with a grain of salt. We then did fluorescence recovery after photobleaching, or FRAP. And we showed that if we photobleached aggregates in the adult, that it didn't recover at all. If we photobleached aggregate-like material, weaker aggregates in the larvae, a little more than half of it actually recovered, meaning that it's diffusible, so it's diffusing back into those centers. And after CRAM1 knockdown, those aggregates are somewhere in between. They basically act a little bit like the hardened aggregates from the control, and a little bit like the incompletely aggregated material or non-aggregated material in larvae. That was L4. I believe it was L4. Okay. So you first start to see aggregates in late L4, and so we were able to see something that looked like aggregates, and we tried to photobleach those spots, but we still saw substantial recovery. So these are the panels you just saw for the 2D gels, and these are comparable panels at day 5, where we've now done knockdown for PQN53 or for CRAM1. Sorry, this is the control at day 5. And what you can see is that this one, CRAM1 knocks down about 98%, and this one about 95% of the protein signal that you saw in the control in the sarcosyl insoluble fraction, the really compact aggregates. In the sarcosyl soluble fraction, the looser, more diffuse aggregates, maybe less cross-linked, you only were able to reduce about two-thirds of the signal, and it was about the same for the two of these. Please don't hesitate to ask any questions. I'm happy to answer questions. Now, one really interesting thing is that David and Kenyon had shown just a year before we prepared these results that aggregates form in wild-type worms as well, and as a function of age. And you can, they identified their aggregates immunologically. We identified them after this same preparation step on gels, and we simply scanned these gels to get the amount of material. And what you can see is that with age, going from 2 days to 5 days post-hatch, you have about a 40% increase in the amount of aggregation with age. And that's true also after CRAM1 knockdown. It's going up, but it's lower in each case. So natural aggregates that are not seeded by a transgene, a transgenic protein, are also inhibited by CRAM1. That's the point of that figure. So we now shifted to a worm model of, I can't say of Alzheimer's, but of A beta aggregation. So these are worms that express human A beta, already 42 amino acids long, in muscle. This is one of Chris Link's strains. And the expression of this A beta leads to paralysis, a loss of motility. And so when we measured the, I'm showing you this is fraction not paralyzed, so what you can see is that roughly 97% of the worms were paralyzed if you just looked at the wild type, which was given feeding vector, not any inhibition. These are worms fed a proteasome inhibitor, MG132, feeding vector plus DMSO, and here's CRAM1, and we've suddenly rescued something around 70%. Well, 70% minus this few percent here. And the interesting thing is that if you simultaneously inhibit proteasomes with MG132, you lose most or all of that benefit. That's not true for PQN22, which is significantly higher than the controls, but it actually increases when you add MG132.
Excuse me.
And if you instead treat the worms with an inducer of proteasome activity, oleuropein, you add something to this, and you get up to close to 80%.
Robert,
That was a different experiment.
Just a quick question. Thank you. Are there any, maybe you've already mentioned this, and I apologize if you did, but are there any phenotypes of CRAM1 deficiency? Do they have?
I will get to that.
Okay.
I will get to that. You're anticipating me, and that's always good. The only problem with the model I just showed you from Chris Link was that it's an inducible model in which A beta is induced starting with a larvae, and it produces within 48 hours of induction as much paralysis as it's going to ever produce. And we really didn't want that to happen because we wanted the aggregates that formed to utilize other proteins that might only be present in the aggregable form, possibly oxidized form, possibly some other form, as the worms aged. So we didn't want this early expression. We wanted late expression as a function of aging. So I talked to Chris about it and said, you know, I'd really like to try them uninduced. Did you ever do that? And he said, no, I never did. So we did. And sure enough, when we didn't induce them, the feeding vector, the control worms, had a paralysis that preceded mortality by a week, or 5 days anyway. But CRAM1 RNAi delayed that by about 30%, and significantly with a p less than 10 to the minus 6. So clearly this can be an aging model if you just allow leaky, low-level expression of A beta to gradually accumulate in aggregates with presumably other proteins. So for this experiment, we wanted to do something a little better than MG132, so we chose an RNAi to one of the catalytic subunits of the proteasome, Pas4. And because we're mixing two RNAis, the effect of CRAM1 RNAi is not quite as great, but when we combine CRAM1 with Pas4, you can now see that you've lost the benefit. So it's kind of like the MG132. If you disable proteasome function, you no longer get the benefit of CRAM1. So CRAM1 somehow is acting through the proteasome, and if you don't have proteasomes, you don't see the benefit. Here we're just showing another way of assessing A beta plaque in these worms using thioflavin T, a plaque-specific stain that has been used in Alzheimer's in situ characterization for years. And you can see with feeding vector only, you see quite a bit of the screen, and if you knock down with CRAM1, you lose about 60% of it, just shown graphically here. So I told you I wanted something that was realistic as a model of amyloid pathology, but there's something terribly unrealistic about what we just did, at least as a model of neurodegeneration, is that the A-beta was expressed in muscle cells, not neurons. So we also had a strain, also from Chris Link, that expresses A-beta in neurons. Now, why not do this immediately? Neurons in C. elegans are small and few in number, and it is rather difficult to count aggregates in those neurons. But they have a phenotype. When you form those aggregates, the worms have a behavioral deficit. And there are actually several behavioral deficits that Chris Link and company have characterized. We chose one that is the loss of chemataxis. So chemataxis is basically, it's not a learning thing, it's an innate attraction to N-butanol, and so we spotted ethanol on this side and butanol on this side and spotted the worms in the middle. And you wait a certain amount of time, 30 minutes, 120 minutes, and you ask how many worms are within a centimeter of this spot, how many are within a centimeter of this spot. And that's the ratio of those is your chematactic index, or it's the ratio of this to the total. So when we used a 30-minute response, we had about 20% of the worms showing proper chemataxis, and that would have been 100% in worms that weren't expressing A-beta. And it just about doubled when we did CRAM1 knockdown. And PQN22 and ATX2 and didn't do much, but PQN53 also rescued pretty well. And the same if we do it at 2 hours, except that now our baseline's 50%. If you wait long enough, half are going to go each way. It becomes random. So we did the same thing about trying to do an aging phenotype. So here the solid bars are day 5, and the stripe bars are day 7. So concentrating on feeding vector, that's what happens without any knockdown. And going between day 5 and 7, you go down from 48% chemataxis to 36% chemataxis. So basically, the worms are aging in their neuronal function. So they're showing a deficit. And if you knock down CRAM1, you rescue both of those time points, but the difference between them stays the same. If you knock down with PQN22, you don't see anything significant, and it's just barely significant at the later time point with PQN53. And then we did some RNAi mixtures so that we could look at PAS4 to knock down proteasomes, and once again, it more than neutralizes the benefit of CRAM1. And again, we're seeing an effect of PQN.
Just a quick question, just to follow. You said that after 2 hours, it's me, here.
You see, okay,
It's
sorry,
me again.
sorry.
It's me again. So after 2 hours, you would expect from wild type to just do 50-50, essentially they would be evenly distributed. So you have a gain of function of CRAM1, so essentially they're preferentially going to...
So what we're saying is that by 2 hours, you would expect, if they've totally lost chemataxis because of the A-beta expression,
not wild type.
they'll be at 50%.
Wild type would be 100 there.
Wild type would still be 100%.
Okay.
Wild type has virtually perfect chemataxis. They just all, it's like, what do they say, bees to honey? I don't know, something. Okay. So we did some in situ immunohistochemistry, and we stained for CRAM1. Okay, on the left is always feeding vector controls. On the right are CRAM1 RNAi in the green box. So the good news is, as we had already established at the RNA level, CRAM1 is pretty much gone except for some the nerve ring in the CRAM1 knockdown. Ubiquitin antibody looks like it gets a little bit stronger, but it wasn't significant. Doesn't change much. And this is the same pairing, but here we're looking at YFP, and you can see as expected that goes down with CRAM1 knockdown because that's what we started with. If we use antibody to ubiquitin, I'm going to try to turn this down now if I can remember which one to do. That says audio. I don't want that. Bright. Is it this one?
I'll do it.
Okay.
My exercise column.
Thank you. Okay. So what happens with ubiquitin is that you lose this punctate pattern, which actually coincides with YFP. But it's a bit weaker. But if you superimpose these, you'd see they're the exact same spots, same pattern. You've lost that with CRAM1 RNAi, but you've got a little bit of, I'm probably blinding you with my laser, but you've got a little bit of background fluorescence. And that becomes an even more convincing story. I shouldn't say even. It becomes a convincing story if you use antibody to proteasomes. So again, YFP going down with CRAM1. Proteasome antibody, you again get this faint punctate pattern that co-localizes with YFP, just as you did for ubiquitin antibody. But can you see all that background now? The background fluorescence has gone up 6 or 7-fold. And this is just a bigger a bigger size magnification of the same thing. But basically, this signal in between the aggregates, it's like the proteasomes were stuck in aggregates and we've liberated them with CRAM1 knockdown. I would forgive you if you jumped to that conclusion, because I jumped to that conclusion. And this is the A-beta staining, and again, it's a similar story in this different strain. When we stain with, for proteasomes, we can see a more intense signal after CRAM1 knockdown, and less or about the same of A-beta total, but much less in punctate bodies. And you can sort of reflect that in the merge, where it's become, relative to here, it's become much brighter in the green relative to the red. So one of my graduate students is from our bioinformatics program, Sundaram Balasubramaniam, and he did modeling of proteins for us, and he showed that CRAM1 has basically an unstructured protein model. It forms 3 rather weak alpha helices. But if you put it together with diubiquitin or tetraubiquitin or monoubiquitin for that matter, it gloms onto it like it was its long-lost brother. Well, maybe brothers don't like each other. Long-lost sister. And he continued this modeling to create some energy calculations. And on the left is delta E, which is called the interaction energy. These energies are negative because you're getting more stable with a negative energy. And so what you're doing is you're basically taking the energy state of CRAM1 plus ubiquitin-1 and subtracting that from the 2 separately. So you're saying this is the change in interaction energy. And delta E reflects the spontaneity of the reaction. How much will this go forward on its own? And delta G is the familiar Gibbs free energy that tells you how stable that interaction is once it's formed. So what's really interesting here is that CRAM1 and either mono or di or tri or tetraubiquitin forms these rather stable complexes, forms these spontaneous complexes, which are also very stable. And here I'm jumping to something that I'll come to a little later. There are human orthologs of CRAM1. There's SURF1 and SURF2. And these proteins also form complexes with tetraubiquitin that are about as stable as the worm one, that are about as spontaneous as the worm ones, and in one case more stable, in the other case less stable. So some other things that you can do with sophisticated modeling software is you can look at some indices of the stable structure of a protein complex. So if you add ubiquitin to sequestosome 1, otherwise known as P62, P62 is kind of like the messenger. It picks up ubiquitin tags on, polyubiquitin tags on proteins, and it guides them to both proteasomes and autophagosomes. And so P62 is known to have a UBA or ubiquitin binding domain, and it does indeed bind. And this is shown here. If you show over time in a GROMACS 10 nanosecond simulation, you can see a lot of up and down movement, and then it stabilizes after about 6 nanoseconds and stays like that forever. On the other hand, this is just showing the radius of gyration from the center of gravity, so to speak. On the other hand, if you toss CRAM1 into the same mix, it never stabilizes. It becomes, if anything, wilder. And you get the same answer if instead of the radius of gyration, you look at the root mean square distance of all the atoms from the mass-weighted center. And again, if you have ubiquitin 4 and P62 sequestosome, the structure gradually becomes more compact. This is stable by radius of gyration, but it's actually declining in terms of structural dispersion, whereas it continues to increase if you toss CRAM1 into the mix. And so here's just a very simple-minded model. I like to create models so that I can convey to you how a truly great mind would envisage this. All right, how I can envisage it, which is not like a truly great mind, but basically a very simple-minded is all you're going to get from me. So here's a misfolded protein. It is recognized and ubiquitinated, and in the presence of CRAM1, you have this CRAM1 glommed onto the ubiquitin. So in the first case, where you don't have CRAM1, P62 basically homes in on it and binds to it. But when you have CRAM1, that's the best I could do, it bounces off, and it's not able to bind. So this is the wild type situation. This is the knockdown situation. And we also used a model of proteasome clearance where you have a transgene constitutively expressed in the muscle cells of the same worm that expresses M-cherry ubiquitin. So it's monoubiquitinated M-cherry, and if UPS activity after E1 and E2 enzymes is normally extending that ubiquitin to polyubiquitin and degrading it in proteasomes, then you should lose the M-cherry because it'll get degraded by the proteasomes. And if that's not working very well, as in the case of CRAM1 RNAi, then you get, I'm sorry, if it's working extra well, you get less of the M-cherry left. And if it's not working well, you have a lot left. So the normal worm is the proteasomes are not working very well at this age, and with CRAM1 knockdown, it is working well. And this is just the aggregates that we knew went down. So if you quantify this, you can say the blue is with CRAM1 knockdown, and you've lost about 40% of your proteasome, I'm sorry, the feeding vector is orange, and this is the M-cherry ubiquitin. So you've managed to lose about 40% of the signal due to proper proteasomal digestion when you do the CRAM1 knockdown. And after 4 days, it's a little bit less of a benefit. So what good is having fewer aggregates? Does it help the worm to live longer? It does. We were the second people to do this because other people had noticed HYPK as the homolog, so they'd also monitored its lifespan in a rather large survey of potential lifespan affecting genes. And they got the exact same answer that we did, which is approximately an 11% increase in lifespan when you do CRAM1 knockdown. So since somebody else did it, we believe it. And why would worms and mammals evolve a protein that, like CRAM1, favors aggregation and reduces survival. That's not what Darwin wrote about, is it? Well, maybe. So the first thing we needed to check was whether developmental rate was affected, and it's exactly superimposable going through the larval stages to the young adult, whether you have CRAM1 knockdown or not. But here's the catch. If you look at reproductive activity, the ability to leave progeny, there's a benefit to having CRAM1 around in the gold color that's not there when you do the knockdown. So at 3 days of age, you're producing twice as many progeny with the feeding vector as after the knockdown. And it's only about a 20% benefit to have CRAM1 at 4 days, and it actually flips by day 5. But here's the point. The total reproductive potential is still about 20% in favor of the worms that have CRAM1. That alone would account for a pretty significant selective advantage for the worms that have CRAM1. So you wouldn't lose it spontaneously. But because this is early reproduction, it's like compound interest. So in fact, the benefit is 300,000-fold over 100 days because you get a lot of reproductive generations, 30 roughly in that 100 days. And so it's this benefit to the 30th power. It's a huge benefit.
So do you know if it's the oocytes or the sperm?
I don't. It's a great question, Pam.
Because you could mate into those.
Yeah, and we should do that. We have not done that. I basically, so I'm trained as an evolutionary biologist and as a breaker of microphones. I got it. And so I was so happy with a satisfactory evolutionary explanation that I didn't want to look any further, but I should. You're absolutely right.
Okay. Robert.
Yes.
So we know that a lot of the time that a worm spends alive is actually potentially in the Dow phase. So any changes on Dow formation?
So we did not, no, we looked and there is no effect on Dower formation at all.
Actually, my guess is that it would be exit from Dower that would be more compromised. Because it's the regenerative capacity when they resume. Yeah, right. They have to clear all the.
And we've never looked at that. So that's.
So look at like something that's been 45 or 60 days in Dower, and they'll recover. I bet.
Write these down. You're obviously intent on co-authoring a paper with me. I think you've contributed enough. Those are good ideas.
Do that. Yeah, it takes you only 3 months.
Only one more question.
Yeah, go ahead.
Is this the same phenotype as the germline knockout?
I'm sorry?
Is this the same phenotype as the germline knockout?
So we also have we got a CRAM1 knockout, and it's very hard to maintain, but it does seem to have the same phenotype. Okay. So just to look at the orthology tree, when you find an orthology, okay, you all know how to find a homolog. You do a blast search, right? And you don't know what the computer's doing, but it's finding matches by basically FASTA algorithms to everything else in the GenBank database. Orthology is a different bird, and it doesn't coincide with homology. So you can have proteins that are not homologous, that are paralogous, and that's exactly what we've got here. So what you do is you look up this lineage to the last common ancestor with these guys, and you say what protein was the common CRAM1-like protein between these two. And then you go to the next step, do it again, the next step and do it again, and then back down to human. And that's a stepwise process that tells you what was identical by descent, what actually evolved from what. And during that evolution, sequences could diverge to the point where you have no significant homology. And so there is some homology to HYPK, but not any at all to SIRF2. And yet, strictly speaking, SIRF2 is the ortholog, HYPK is not, because HYPK is a partial duplication of SIRF2 that occurred late in evolution, only in the mammalian line. But interestingly, here's SIRF2, here's HYPK, and they are co-transcribed as a splice transcript. So the fact that that exists in EST hits from GenBank tells you that these are on the same operon. These are co-transcribed. So I use that as an argument in this aging cell paper that these had to co-evolve together. If they're co-transcribed, then it may be that selective pressure on one affects the other. It's a weak argument, but it's the best I had. So first part, summary and conclusions, and this is the part that's published so far, okay, plus some supplementary stuff. Proteins discovered by aggregate proteomics or aggregomics may play pivotal roles in aggregation. They're not just all innocent bystanders that got stuck in the tar pit, but they may be things that contribute functionally to aggregation. CRAM1 and its mammalian orthologs may represent, this was the best I could come up with at the time this paper went to press, may represent a primitive novel class of chaperones that favor the reproductive state, but during aging interfere with proteasome function. So if you think about it, as an animal is going through its early adult development when it needs to reproduce, proteins will still aggregate, especially if it encounters a hostile environment with lots of oxidative stresses. You'll get lots of oxidation. All evolution cares about is that that worm reproduces and lives long enough to reproduce. It doesn't give a darn whether the worm lives after that. And so one or more such proteins, CRAM1, maybe SERFs1 and 2, form stable complexes with ubiquitin and with oligoubiquitin. And I'll show you in a few minutes that they also form stable proteins with lots of stable complexes with many of the other proteins that were in the aggregates that we isolated. And they block critical interactions with p62 proteasomes and autophagosomes. These are the people who did the work. Mainly my close colleague and associate Srinivas Ayadavara. This is Sundaram Balasubramaniam, here before he got a haircut, here after he got a haircut. He's the very talented bioinformatics graduate student. This is our technician, Ramni Allah, and some other people. There's me. And we're supported by the Veterans Administration and by the Life Extension Foundation and even the SENS Foundation. So when you get desperate for NIH funds and they're not coming, you turn to smaller dollops of money and they keep you going. So this is the end of the part that's published data. I'm going on to unpublished data now. So I already showed you that CRAM1 has 3 alpha helices, and what I'm showing you here is this is CRAM1. This is a consensus UBA domain, ubiquitin binding domain, from a variety of mammalian proteins. And they have a very similar structure with essentially the same arrangement of 3 helices. And if you align these UBA domains, sorry, the scale is different. This is CRAM1. It's a short protein. It's only 96 amino acids long. But it has this 40 amino acid long UBA domain. And sequestosome 1, also known as p62, has the same domain at its C-terminal end, but it also has a PB1 superfamily and a ZZ superfamily, zinc finger superfamily upstream here. RAD23A from mammals has a ubiquitin UBQ superfamily, which helps to guide the ubiquitin tag protein to subsequent degradation, and it has 2 UBA domains, one of which is right at about the same position as in sequestosome. So the interesting thing here is that a recent paper on RAD23A, they actually truncated the RAD23As, breaking it right about here, so that it had this UBA domain like CRAM1, but no longer had the targeting part, and it now, in fact, disrupted aggregate clearance. So it functioned just like CRAM1. So basically, the next logical experiment for us to do was already done, which is nice. So here's the RAD23 N-terminal, sorry, C-terminal UBA domain, and this is CRAM1. And here is a plot of interaction energy. So this is the initial spontaneity factor, the delta E, not delta G, of formation of a complex between CRAM1 and a set of proteins that we found in its aggregates. And I'm just going to give you a little heads up. Many of these are proteins that we will show you in a minute also were found in aggregates purified from Alzheimer's, human Alzheimer's samples. So 1433, both gamma and eta, they're the same two isoforms that appear in Alzheimer's. DNJ11, PGAM5, HSP70, both of these appear in Alzheimer's, not DNJ11 so far. A neuropeptide. These all are going to rush into aggregates with CRAM1. They love CRAM1. They're enamored of one another. Q40YFP, which is what we really use to seed the aggregates, also works, of course. Diubiquitin is about as good as Q40YFP. And PQN53 is not far behind. This is the other neuropeptide in LP29. Heat shock factor 2 is not a bad contributor. This should be ATX2, which is the ataxia gene. And another dozen that I only show 2 of here had no interaction at all, which is kind of what you'd expect.
This is all model data?
So,
This is all model data?
so this is done in GROMACS modeling the interaction in solution.
So you had structural data on these proteins?
No, so Sundaram has actually created a proteomic suite in which he brings together a lot of software developed by other people and automates its execution for a series of proteins. So he's made what are called bash scripts. But what he can do with this is run GROMACS, which is a dynamic molecular modeling program for each of these pairs of proteins in sequence.
So they must be homology models.
And he's tied this to either the crystal structure, if it exists, which he then improves with what's called a threading model. And if it doesn't work, he uses just the threading model. And the really interesting thing that you need to know is that when he did the threading model for proteins for which crystal structure was already known, 12 out of 12, he got the exact same answer.
Yeah, but it would have been threaded on a crystal already.
No, I'm saying without using the structure, he got the same answer as with using the structure.
So de novo homology modeling.
Yes.
Nice.
So that was not true just a few years ago, but the accuracy of these models has incredibly improved. And it is, in fact, GROMACS that got 3 people the Nobel Prize 6 months ago or so. So the other thing that he also incorporates is what is sometimes called evolutionary conservation modeling, where you line up all the related sequences in nearby species and you look for these conserved residues. And those conserved residues are conserved for a reason. And the leap of faith is that that reason is structural. We don't know that, but that seems to work. Okay. So this is just showing the complex with tetraubiquitin on the left with the yellow part, which in this case is CRAM1 UBA domain, and in this case is the RAD23 UBA domain. Not much difference. Here he's shown some other complexes of other proteins shown in blue with tetraubiquitin. So again, all the stuff on the left is 4 ubiquitin copies, and the blue part here is P62. Here it's P62 again, but just switching to the C. elegans, tetraubiquitin. I don't know why he did that. And this is HYPK over here with ubiquitin. So looking at just the interaction energies of the UBA domain, so you actually delete everything but the UBA domain, it turns out that CRAM1's UBA domain is about as good as any of them get. HYPK is the only thing that's maybe a little bit better. RAD23 is actually a shade worse, on a par with P62 and the human UBA consensus sequence. So you're getting these fairly strong spontaneous interactions of just the UBA domains with ubiquitin. Note that these are not as negative as the whole protein, which was around minus 250. So there are other interactions that stabilize those proteins when they get together with other proteins. But those are mostly electrostatic and hydrophobic interactions. So this is a technique called proximity ligation amplification, or PLA, in which you can test whether things are close to each other that you think are close to each other, because PLA requires that the two things you have antibodies to are within 30 to 40 nanometers of one another. So if you do that with proteasome and ubiquitin, of course that works. It gives you a glow everywhere. If you do it with CRAM1 and ubiquitin, you get a fainter signal if you knock it down, but you get a pretty strong signal here, showing that CRAM1 really does interact with ubiquitin, as we had modeled and found. So this is just a repeat experiment showing a quantitative knockdown with CRAM1 at a rather early time point, just the number. It's not as strong a knockdown as we usually get, but just to show again that if you inactivate the proteasome with MG132, you lose that benefit. And here he used RNAi to ATG7. And the interesting thing there is that ATG7 encodes the ubiquitin activating enzyme E1-like protein. So it starts the activation before any conjugation occurs of ubiquitin for conjugation. So if you can't get ubiquitin on, ATG7 disruption is blocking the benefit, the blue-red difference with CRAM1. Whereas LGG3 and Beclin-1 don't block it at all. Now, having said all this, we have not yet tested these for efficacy of knockdown. So this is all hot off the press and subject to confirmation. But the interesting result that ATG7 does this becomes more interesting since I just learned today that in fact ATG7 is, I think it was Al was telling me, ATG7 is much more autophagy associated than it is proteasome associated, although I had thought it would, if you knock it down, you block all ubiquitination, so it should affect both. But what do I know? This is a new area for me, and so I defer to your greater wisdom. Okay, so this is now using ubiquitinated M-cherry to look for the clearance of this structure. And so, again, when you knock down CRAM1, you get improved clearance, and that's not blocked if you knock down CRAM1 and knock down LGG3 and knock down Beclin. But same story, if you knock down ATG7, you completely block, or if anything, reverse the improved degradation through proteasomes. And this is just showing the aggregate count when you knock down CRAM1 diluted 50-50 with feeding vector, or knock it down along with RAD23, it reverses that. So it's same sort of story. So I'm almost embarrassed to show this slide because it will undoubtedly reveal what I like to call untapped depths of ignorance on my part. This is not an area that I know well, but I wanted to present, just in case there's anyone in the audience who knows the area less well than me, I will now proceed to confuse you. So the ubiquitination pathway starts with the E1 type enzymes, such as, I thought, ATG7, and that activates the ubiquitin, monoubiquitin, which is then conjugated by the E2 enzyme for addition to a protein, and then the E3 ubiquitin ligase extends that. And so you end up with polyubiquitin, and when it's about 4 or 5 long, that's enough for it to be conducted by P62 sequestosome and other complex members to proteasomes, which I've rather crudely shown here as the proteolytic subunits and the cap. And RAD23 I drew in the cap, although I gather it's only partially there. But again, my not quite so brilliant conceptualization of this was that the things that if you block CRAM1 knockdown doesn't work anymore are putting on the ubiquitin and recognizing the ubiquitin, because that's what RAD23 is alleged to do in the cap. If the proteasome doesn't work, what do? Who are you going to call? Not Ghostbusters, but autophagosomes. So when you go to autophagosomes, this is where LGG3, which is also is the ortholog of mammalian ATG12, along with APG7, APG5, and with Becklin-1 ATG6 mediating vesicle fusion through the type 3 PI3 kinase. Autophagosomes are late, and nothing that we did down here had any effect on the CRAM1 benefit. I would not infer from that autophagy is not important to aggregates in C. elegans. They've got nothing but autophagy to eliminate aggregates. You can't do it with proteasomes. They won't. They just choke on it. It gets constipated. Okay, they can't get through here. So they depend on autophagy. But the point is that CRAM1 acts earlier. So there's a proteasome defect that is the heart of where CRAM1 acts. That's all that I'm trying to say, not anything about the relative importance of these two pathways. They're both important. So second summary and conclusion. Protein aggregates form because many proteins don't have a tightly defined structure. They hover on the knife edge between stability and instability. And when they're damaged and misfolded, they can be sticky. They can expose their normally internally sequestered hydrophobic residues to the outside. That apparently also follows oxidation many times. Or they can expose allosterically a phosphorylatable site that changes the structure and obviously the charge of the protein. So CRAM1, SURFs1 and 2, and HYPK are sticky by design. That is, they seem to cohere to a lot of proteins, but especially well to ubiquitin. And depending on what other domains they contain, they may help to target aggregate proteins to proteasomes and autophagosomes, like sequestosome 1, or just to one or the other. And then lastly, CRAM1-KD requires proteasome function. So if we knock out proteasome function with RNAi to one of the subunits or with MG132, then we don't see a CRAM1 benefit. It requires ATG7 and E1 ubiquitin activator to allow tagging, and RAD23, again, as the Pope would say, I'm fallible on this one. RAD23 is for ubiquitin tag recognition at the proteasome. So at this point, you're muttering to yourselves, at least the non-C. elegans people in the audience are muttering to yourselves, okay, but these are worms. They can't forget where they left their car keys. They can't even drive cars. And they don't get Alzheimer's or Huntington's. So what about human neurodegenerative diseases? Well, we thought the same thing. So we have been studying Alzheimer's brain samples from the hippocampus. The posterior or caudal hippocampus is the main affected region histologically in Alzheimer's. And we are also beginning work with Parkinson's affected regions. So I'm going to summarize some of the data here, but let me just tell you, we did the same kind of aggregate isolation and purification using magnetic bead pull down either to A beta or to tau, total tau, not just phosphorylated tau. And what this very complex diagram shows is on the left, the diamonds are A beta aggregate proteins, and the red is always the Alzheimer's compared to the age-matched controls. So the up arrow means that this shifted upwards, and this is on a log 10 scale. So that's a sizable shift, but it's an even bigger shift for tau. And so all these proteins that I'm showing you here are ones that shifted for one or the other. But, bless you, the truly remarkable and unforeseen thing, unforeseen by most people, not by me because I had my preconceptions, but is that most of the proteins that increased in frequency with Alzheimer's disease in the A beta aggregates also increased in the tau aggregates, even though these are meant to be totally geographically separated. And I'm going to show you that's not true. So the Alzheimer's A beta aggregates are in plaque, which is mostly outside of neurons, and the neurofibrillary tangles are in the neuron projections called neurites, and it's thought that the two don't meet, but there actually have been a flurry of papers in the last year indicating that A beta does occur inside cells and maybe is most important when it's inside cells. But anyway, we got some interesting protein hits here, and a lot of coincidence between the two kinds of aggregates. And notice that we've got things that you've seen before, like HSP70, which also appeared for the C. elegans aggregates, 1433 beta, alpha, eta, gamma, et cetera. Those all were shifted. Tenacin we also found with C. elegans. Sequestosome 1 we also saw with C. elegans. These laminins we didn't see. Tau we didn't see, but tau barely shifts with which one is which? In the A beta aggregates, and it doesn't really shift with the tau aggregates. It just gets phosphorylated is the main change that we had. It doesn't get into the aggregates more. It's getting phosphorylated a lot more, and the phosphorylated form is in the aggregates more. So we wanted to do a functional test, and we turned back to C. elegans, where we can address the importance of each of these proteins the same way we did for CRAM1. So we knocked down in two different A beta expression models, the two I showed you for movement, paralysis, and chemotaxis. And on this scale, everybody's normalized to 1. So if there's no change, the controls are 1, okay? And everybody here is pretty much up, but not always significantly. So if you see the stars, it means it was significant. So these two 1433s were significantly elevated in one or the other system. Alpha enolase is significantly elevated in both systems. Amyloid beta APP protein is significantly elevated. Just go right through the list. Band 4.1-like protein is activated, clathrin, GST, HSP70, lots of changes. And the most profound ones include tau for a functional effect, laminin gamma 1, and tenacin receptor. So we got some really interesting hits. And the question is,
Those are knockdown?
so these are all looking at the phenotype after knockdown. And we start the knockdown at the L4 adult mold so that we're not affecting development, because many of these would have deleterious effects on development. So we used PLA to ask, and also just immunohistochemistry to ask the question, are these things that we have now inferred are co-aggregating together, are they really in contact in the cells in Alzheimer's? So on the top we have Alzheimer's plaque. Here we have a similar part of the cortex for age-matched controls. You see tau in green. In this case, it's phosphotau, so there's no phosphotau in the age-matched control. There's a lot of it in Alzheimer's. And when we looked, red is dynactin, and we pulled down dynactin with antibody to tau, so we said those two should be in the same place. And when we did the PLA dual link, we got this yellow signal only in Alzheimer's, not in age-matched controls. Please don't groan. I know this is a tautology. If there's no tau, how can you have tau interacting with anything? But remember, this is phosphotau that we used antibody for. There is tau here, it's just not phosphotau. Here, this is just immunohistochemistry for 3 different Alzheimer's patients looking at their hippocampus. I think the plural is hippocampi. And we did this also for age-matched controls, and we saw nothing. But green is phospho-tau, blue is dynactin, and what you're mostly seeing is blue-green. So that in fact, you're seeing these in the same place. And where you see red, it doesn't co-localize. That's the APP. So it's not, we didn't know that if we could see the co-localization. We predicted a little bit, but we didn't see it. So we concluded that A beta is maybe in proximity to some total tau, but only in AD hippocampus, not in AMC. And we tested that by doing PLA between tau in red. This red is the signal for the PLA, for proximity ligation amplification, of looking for tau and A beta within 30 to 40 nanometers. And I hope you can all see these red clusters that are in the neurite projections. And we did counterstains to show that. They just don't show up here. And this is age match control. There's still tau. So this is incorrect. This is not phosphotau. This is just total tau. So we do see total tau in about the same amounts, but we don't see the interaction with A beta.
Robert, just curiosity, what was the epitope for the antibody, the A beta antibody?
Say again?
Do you remember what the epitope was? Was it an N-terminal antibody? Was it a pan A beta or?
So the antibody to total
A beta.
To A beta or to tau? Are you talking about to tau?
No, A beta. So which one was it? The the isoform specific?
This experiment we used Mary Jo Ledoux's A beta 1 to 42 specific antibody that does not cross-react with APP.
I see. Okay.
So it's the only antibody that you can say that about. Regarding the tau, it's an antibody to total tau that is monoclonal, but it's meant to interact with all the different tau isoforms. The reality, and this is something that between now and publishing this I may conveniently forget, no, I'll remember, is the phosphotau is mostly turning up the isoform that is phosphorylated in Alzheimer's. It was billed as being general phosphotau, general antibody to tau, total tau. But in practice, when you make an antibody, it is what it is, and it turns out that if you actually check, it's reacting mostly to the isoform that is in Alzheimer's.
If you run this same series to try to see the closeness of interaction on what's considered to be a completely tau-related disease, do you end up with the same results, or has anybody done that?
Nobody's done that. That's a great question.
Frontotemporal.
Yeah, no. We need to do that. But understand that because this antibody for A beta 1 to 42 is absolutely specific and does not recognize APP, all neuronal cells make some APP. They need it, presumably for memory. They don't make A beta except in Alzheimer's. They make some, but not the 1 to 42. They don't make much of. That's really a function of the gamma secretase, right? So you're going to see that in Alzheimer's. So I don't expect there'd be a positive, but I'm going to look now that you mention it. It's a good idea. And that was it. So I could have told you even more stuff. I didn't realize that time was going to fly so much, but I will be happy to take further questions. Anybody?
Question.
Yes.
So you said that the human homolog of prime 1 is SIRF1, right? So
the closest homolog is HYPK. It's got predominantly N-terminal homology that reaches an E value. If you did a blast search, it reaches an E value of 10 to the minus third or something. It's barely worth mentioning. But it's not any better homologous to anything else.